Dear learners, today we are going to discuss the MCQs from Microbiology, the Part 2 from the topic Bacteria. In Part 1, we have covered a few of the MCQs from Bacteria. Now, the first question from Part 2 is enzyme hydrolyzing bacterial cell wall is. Now, which of these enzyme hydrolyzes the bacterial cell wall? Whether it is amylase, whether it is reductase, whether it is protease or lysozyme. The correct answer which is lysozymes which hydrolyze the bacterial cell wall. This lysozyme is responsible for breaking the bond, that is 1,4-beta linkage between NAM and NAG, that is N-acetylmuramic acid and N-acetyl glutamic acid. And this linkage, this bond and this linkage of NAG and NAM forms peptidoglycan, which is a major component of gram-positive bacteria. So, since lysozymes are the enzymes which are responsible for breaking this bond, will not allow this bacteria to form the cell wall, this peptidoglycan layer. So, these lysozymes are responsible for hydrolyzing the cell wall, and because of the property of breaking the bond, that is 1,4-beta linkage, these are also known as N-acetylmuramide glycan hydrolase. The next question is, the site of energy production in a cell is, whether it is microbody, whether it is chromosome, whether it is ribosome or mitochondria. The correct answer is, it is mitochondria. The energy production house of a cell is mitochondria. As you all know, mitochondria is called as the powerhouse of cell. Now, why? Because this mitochondria contains many enzymes for carrying the Krebs cycles or citric acid cycle. Now, this citric acid cycle or Krebs cycle will produce NADH, that is nicotinamide adenine dinucleotide, and the reduced form is hydrogen, that is NAD is nicotinamide adenine dinucleotide and H is reduced form, responsible for the utilization. Since the name ATP synthetase, it will synthesize the ATP. The next question is, bacterial ribosomes are composed of, whether they are composed of protein and DNA, whether they are composed of protein and mRNA, whether they are composed of protein and rRNA, and whether they are consist of protein and tRNA. Now, you might find all these options resembling, but the question you will have to focus now on question bacterial ribosomes. Composition of ribosome they are asking. So, first we will see what are ribosomes. Now, as you can see, this is the larger unit of ribosome, this is the unit of ribosome. mRNA attaches and this is the transfers RNA and this is the growing polypeptide chain. So, mRNA, rRNA, this mRNA, tRNA, these are the other parts, but which are also essential for the protein synthesis. But the own composition of ribosome says that it contains rRNA, that is ribosomal RNA, and proteins. So, the correct answer is the bacterial ribosome is composed of protein and rRNA. Next question is, diphtheria is caused by which of these species? Corynebacterium, Staphylococcus, Streptococcus or none of these? The correct answer is, it is, since diphtheria is caused due to Corynebacterium diphtheriae. So, we will see now what Corynebacterium diphtheriae is responsible for. It will produce a toxin, exotoxin, which will cause a membrane of dead tissue, as you can find here, to build up over the throat and tonsils, making breathing and swallowing difficult, which you can note here. So, this can be due to bacterial growth, due to a toxin and exotoxin production, due to necrosis or due to host immune response. So, it is caused due to Corynebacterium diphtheriae. Next question is, the most infectious foodborne diseases, whether it is tetanus, whether it is dysentery, whether it is gas gangrene or botulism. The correct answer is, it is botulism. infectious foodborne disease is botulism, most infectious. Why? Because botulism is responsible for producing toxin, that is Botox, which is a neurotoxic protein produced by bacterium Clostridium botulinum and other species of Clostridium botulinum. And the symptoms, as you can see, these is main symptoms: nausea, vomiting, then facial weakness and difficulty in swallowing, etc. So this is when these toxins are produced. These toxins enter our body through food and they will cause botulism. So the correct answer is, it is botulism, the most infectious foodborne disease. Next question is, acridine dyes are more effective against, whether they are more effective against gram positive, gram negative, Rickettsia or Mycoplasma. So as you all know, acridine as you all know, which is a famous moiety present in an antiseptic disinfectant, that is proflavin. Now the mechanism is, it is mutagenic on DNA, and since it is responsible for causing the mutation, it will prevent the bacterial reproduction. It is having the mechanism that intercalates between the nucleic acid base pairs by insertion or deletion, and the normal mechanism of intercalation is substitution. So this will produce a double bond stranded breaks in the DNA, and it will prevent the reproduction of bacteria. So the correct answer is, it is gram positive bacteria. Acridine dyes are more effective against gram positive bacteria. Next question is, the procedure of differential staining of bacteria was developed by AH gram, HC gram, NC gram or HA gram. The correct answer is, it is HC gram. The procedure for differential staining was developed by the scientist called Hans Christian Gram, that is HC gram, Hans Christian Gram. So it is also known as gram staining, which is a part, or which is a kind of gram staining, means it is a part of. Differential staining is further subdivided, and the divisions are gram staining, capsule staining, spore staining, etc. The next question is, bacillus is an example of, whether it is an example of gram positive bacteria, gram negative bacteria, virus or viroid. This bacillus is an example of gram positive bacteria. The correct answer is gram positive bacteria. Next question is, amoebic dysentery in humans is caused by. Now as you all know, amoebic dysentery, it is caused due to a protozoan parasite, which we have seen in anti-amoebic drugs video. So amoebiasis is also known as amoebic dysentery, which is caused due to a protozoan parasite called as Entamoeba histolytica. So Plasmodium, Paramecium, yeast, Entamoeba histolytica. The correct answer is, it is Entamoeba histolytica. Amoebic dysentery or amoebiasis is caused due to Entamoeba histolytica. Next question is, mordant used in gram staining is. Now gram staining procedure, which in short we have discussed in part 1. According to that, mordant used in gram staining is, whether it is crystal violet, iodine, safranin or all of these. Crystal violet cannot be there because it's a primary stain. Iodine, not sure. Safranin is also not the correct answer because it is secondary stain. So the possibility is iodine, because the last option is all of these. The correct answer is, it is iodine. This mordant is having the major function that it works as a dye fixator. Means iodine will fix the primary stain, that is crystal violet, to the gram positive bacteria, and due to that fixation, due to the complex formation, this decolorizer will not be able to decolorize the gram stain, and since because of this color fixation or dye fixation property of the mordant used, gram positive will retain the primary stain. Next question is, gram staining is an example for whether it is an example of simple staining, differential staining, negative staining, or none of these. So according to the types of staining techniques are divided into simple staining and differential staining. Simple staining, that is monochrome staining, makes the use of single stain, and differential staining is generally used, as the name suggests, differential. It is used to differentiate. So whether the identification is concerned, gram staining and acid fast staining will identify that whether the bacteria is gram positive or gram negative, acid fast or non-acid fast. And visualization of structure, in that two differential stainings will come, spore staining and capsule staining. So as the name suggests, visualization, they are used for visualizing these structures, spore and capsule. So gram staining, according to the staining techniques classification, it is an differential staining, because as the name suggests, differential, it is used to distinguish between gram positive and gram negative. So the correct answer is gram staining is an example of differential staining. Next question is, a facultative anaerobic is that who only grows anaerobically. Anaerobically means in absence of oxygen. Only grows in presence of oxygen. No, only presence of oxygens are aerobics. Only anaerobics, so it is the term that is facultative anaerobic. So first also it is not possible. Then third is ordinarily an anaerobe but can grow with oxygen. No, ordinarily it is an anaerobe but can grow with oxygen. Yes, it is an anaerobe, it can also grow with oxygen, but only when it is actually an anaerobe. It is actually ordinarily an aerobe but can grow in absence of oxygen too. Now it is the correct answer for this question is, it is an ordinarily aerobe but it can also grow in absence of oxygen. The question is, the percentage of oxygen required by moderate anaerobe is. Moderately anaerobic means they require moderate quantity of oxygen. Moderately anaerobe means anaerobes are which grow in absence of oxygen, but moderate anaerobe means a little bit concentration of oxygen can be tolerated. So what is that concentration? 0%, less than 0.5%, 2 to 8%, or 5 to 10%? The correct answer is, it is 2 to 8%. Moderate anaerobes require oxygen in concentration of 2 to 8%. Next question is, color imparting agent in staining is, whether it is mesosome, plasmid, mitochondria or chromophore. The correct answer is, it is chromophore, because chromophore at a particular wavelength it absorbs the light, and since they catch or absorb the light, chromophores are responsible for imparting the color. That means they can steal the color from the color wheel. So the correct answer is, it is chromophore. And the another part of stain is oxochrome, which imparts the ionization properties. So the correct answer is, color imparting agent in staining is chromophore. Next question is, spirochete is a gonococci, staphylococci, treponema pallidum or streptococci. The correct answer is, it is treponema pallidum. Spirochete is treponema pallidum. And other spirochetes, as you can see, which are responsible for human disease or associated with human diseases, are treponema pallidum, which is responsible for disease syphilis, which is a sexually transmitted disease. Then Borrelia and other species are responsible for various epidemic and endemic fevers. And Leptospira is responsible for a very well known disease, leptospirosis. So these are the other spirochetes which are associated with the human disease, and the correct answer is, it is treponema pallidum. With this we finish with few of the MCQs from the topic Bacteria Part II. In Part 3 we will cover few more MCQs from the same topic, that is bacteria. So stay tuned. Thank you.